microbiota, hepatic fibrosis, and hepatocellular carcinoma. It's not working, sorry. Oh, okay. Some information was presented before, so I only add some more information. Average weight of microbiota is about 1 kilogram, and it's composed of more than 1,000 different species, and the number of genes in 100 to 150 times more than the human genome, and it means that it's 10 or 15 times more cells than the whole human body. The total microbiota is composed of many phyla and species, but almost 90% of human microbiota is composed of two main phyla, Bacteroidetes and Firmicutes. And as you can see, the microbiome is very rapidly evolving in first months of life, and it is stable at the age of 2 years old, but everything depends on genetics, but we also must remember that this microbiota composition is affected by some life events, and as you can see, first of all is type of feeding, but also the drug administration, especially antibiotics, and of course malnutrition, some metabolic diseases such as obesity and type 2 diabetes, and finally microbiota can be changed in the older age. Microbiota has many functions, but basic function is the protection against pathogen colonization, maintenance, and stimulation of immune response. Some metabolic function, first of all, supply of additional calories and energy, and production of short-chain fatty acids and some kinds of vitamins. We must remember about drug metabolism and carcinogens degradation, and also some regulation in intestinal motility and maturation, stabilization, and proliferation of enterocytes and colonocytes. Short-chain fatty acids are very important in our life because they are an additional source of calories, but we must remember that some of these short-chain fatty acids, especially butyrate acids, also is important in the regulation of metabolic pathways, and it's the one of main regulators of insulin resistance because it can increase the amount of incretin hormones, mainly GLP-1. Microbiota also plays a pivotal role in regulation of immune system. As you can see, the surface of small intestine exceeds 100 square meters, so the proper microbiota is the first main barrier protecting against infection, and both specific and non-specific immune responses are involved in defense against pathogenic microbiomes. And all these immune responses are regulated by pattern recognition receptors, which include two kinds of receptors. First of all is nucleotide binding oligomerization domain-like receptors called NOD-like receptors, which are related to inflammasome and localized in cytosol. And the second type are toll-like receptors, which are localized on the surface of cells. Activating factors for these receptors are pathogen-associated molecular patterns. It means some products which are exerted by bacteria, and the main driver is here endotoxin lipopolysaccharide. But also these factors are endogenous damage-associated molecular patterns, which means fragments or died or disintegrated cells, mainly hepatocytes. All these receptors are localized on leukocyte, dendritic cells, macrophages, NK cells, sinusoidal endothelial cells, hepatic stellate cells, and hepatocytes. And as you can see, these inflammasomes are a specific intracellular protein complex acting as innate immune system receptors and sensor that regulates the activation of caspase and induce inflammation response to infectious material. And we can see that activation of inflammasome is very, very high in patients with non-alcoholic fatty liver disease, especially with NASH. It's even higher than in other chronic liver disease, such as hep C infection. Here, these slides show how lipopolysaccharide and high fatty diet increase endotoxin levels. On the other side, we can see that inflammasomes, lipopolysaccharides increase the pro-inflammatory response and pro-inflammatory cytokines expression, but not only in the liver, but also in subcutaneous and visceral fat and muscles. And in normal states called eubiosis, microbiota is characterized by diversity, richness, and relative abundance. And we must remember that microbiota and host live in a cooperative systemic aggregation model. But this microbiota must be kept under control. And to control this microbiota, we need mucosal barrier integrity, secretion of proper amount of gastric acids, biliary salts, pancreatic enzymes, activation of local and systemic immunity, intestinal motility, and interaction among different bacteria species called quorum sensing. Here we have this proper gut barrier, and proper gut barrier maintain homeostasis between different metabolically active tissue. But what is happening when we have some abnormal condition called dysbiosis, which is connected with downgrowth, overgrowth, and outgrowth of bacteria? We have the problem, we have the leaky gut, and this leaky gut is due to the loss of tight junction. This loose tight junction enabled or facilitates the passage of bacterial fragment and toxin from the lumen of the gut to some other tissue, especially to liver tissue and adipose tissue. And this passage evoke adipose tissue inflammation, hepatic steatosis, hepatic inflammation, hepatic fibrosis, and increase insulin resistance. In normal condition, adipose tissue is very silent. It's composed of small adipocytes and some stromal cells. But in inflammatory states, these adipocytes enlarge, and also there are many, many stromal cells with pro-inflammatory polarization. All this condition leads to increase of pro-inflammatory cytokines, higher lipolysis, adipose tissue insulin resistance, but also the higher release of hepatotoxic leptin and the lower amounts of hepatoprotective adiponectin. All this condition leads to sublime and to higher amounts of free fatty acids, which are collected in hepatocytes and leads to hepatic steatosis. In insulin-resistant states, we have not only metabolic problem, but insulin resistance also activates some other pathways, especially it activates cell proliferation of survival due to inflammatory states, and high insulin resistance provoke reactive oxygen species and oxidative stress, which destroy DNA and finally provoke oncogenesis. In the compensated insulin resistance, we have another problem. We have very high amounts of insulin, and we know that insulin have two activities. First of all, if metabolic activity associated with glucose metabolism, but the second pathway of activity of insulin is pro-proliferative and pro-proliferative activity. And in this state, we have higher cell proliferation and also higher cell growth and survival. And in condition of inflammation, we have higher possibility of neoplastic transformation. Finally, leptin. Leptin is one of the best known adipokines, and you can see that it's not very good adipokine, especially in this case of inflammation. You can see that this adipokines aggravates fibrosis and accelerate inflammation. And all these conditions, together with action of leptin, also increase the risk of hepatocellular carcinoma due to the increase in the rate of angiogenesis and stimulation of some endothelial cells proliferation. Angiogenesis, as was shown in the study by me and colleagues and Professor Hartleb, showed that angiogenesis was shown at the very early stages of chronic liver disease, especially non-alcoholic liver disease, even without any fibrosis. So it was very surprising results, but it means that the cancerogenic potential is started at very early stages of non-alcoholic fatty liver disease. And here you can see that these new pathological blood vessels are mainly located in the area of steatotic hepatocytes, and they are very abundant in other places of the liver, but mainly was shown in the area of steatotic hepatocytes. We must remember then in patients with non-alcoholic fatty liver disease, there is different ratio of some phyla and species of bacteria. You can see that in patients with obesity and NASH, there is higher amounts of Bacteroidetes with lower amounts of Firmicutes. And there is also shift to a higher amount of Proteobacteria and Verrucomicrobia, which degrade mucus and promote bacterial translocation. Additionally, Proteobacteria produce large amounts of hepatotoxic endotoxin, and finally, some of these bacteria can produce endogenous ethanol, which additionally exert hepatotoxic action for the liver. We must remember that leaky gut is associated with higher permeability, and as you can see here, this bad gut barrier and higher permeability of the gut was shown in almost in all patients with moderate and severe fibrosis. And bacterial dysbiosis, small intestinal bacterial overgrowth, was observed in 90% of patients with moderate or severe fibrosis compared to just 10% of patients with minimal or mild fibrosis. This bacterial translocation and passage of lipopolysaccharides activates some inflammatory cells in the liver. First of all, its macrophages are activated by the toll-like receptors. These toll-like receptors activate these cells to produce some pro-inflammatory cytokines. These pro-inflammatory cytokines cause hepatocyte injury, but first of all, activates hepatic stellate cells to produce collagen and aggravates fibrosis. What is important, they are also important on the influence of inflammatory process. They decrease the count of NKT cells, which are first line of our fight against potentially neoplastic cells. These cells are able to destroy these pre-neoplastic cells, but when we decrease the number, there is the higher risk of neoplastic transformation. As you can see here, endotoxin not only increase hepatic steatosis, but also increase the toll-like receptors expression on the surface of the cells and increase fibrotic area, collagen production, and smooth muscle cells activity. In inflammatory states, we have a phenomenon which is called Lipo Toxicity. This is dysregulation in lipid environment and lipid composition leading to accumulation of very harmful lipids, which leads to some dysfunction of endoplastic reticulum and mitochondria and cause cell injury and death. And you can see that one of these harmful lipids are ceramides, which a few years ago was treated only as a structural lipids, but now they are also very important in metabolic regulation. Generally, we can divide risk factor of HCC as into viral and non-viral risk factor. And when we look at geographical distribution of cancer risk factor, we can see that oriental part of the world, I mean Asia and Africa, are still we still observe the viral risk factor. But when we look at Europe and North America, we can see that metabolic risk factor consists almost 50% of cases. This nice study show how the role of metabolic risk factor increased in England in patients qualified to transplantation due to liver cancer. We can also see that not only non-alcoholic fatty liver disease, but other ingredients of metabolic syndrome are higher and higher in patients with liver cancer qualified to liver transplantation. What is more important is the fact that almost 50% of cases of HCC in patients with non-alcoholic fatty liver disease is observed in patients without significant or even any fibrosis. So we don't need fibrosis to develop the liver cancer in patients with non-alcoholic liver disease. Liver disease, non-alcoholic fatty liver disease, is strictly associated with type 2 diabetes, and you can see that in type 2 diabetes there is higher risk of many cancer, but the higher risk is for the liver cancer. Also, the obesity and overweight is associated with higher risk of many cancer, but the highest risk is for the cancer of the liver. One of the factors which is also associated with risk of HCC in diabetes is dysbiosis. Dysbiosis is in patients with obesity and type 2 diabetes means that it's low diversity of microbiota or low gene count. And this low gene count is associated with profile with pro-inflammatory activity, reduced number of bacteria which produce butyrate acids, and this butyrate acid is responsible for good gut barrier. And there is also increased number of bacterial species responsible for mucus degradation and decreased number of bacterial species responsible for colonocytes and enterocytes regeneration. So what is the connection between microbiota, dysbiosis, and HCC? We have very, very scale data up to now, and there is only data on animal models. There are no data on human. So you can see that the depletion of host microflora after antibiotic treatment can suppress tumor formation with significant reduction of the number and size of HCC nodule in mice. Mice growing in germ-free condition develop fewer and smaller HCC than conventional mice, and chronic treatments with a low non-toxic dose of lipopolysaccharides led to significant increase of the number and size of HCC. And modulation of dysbiosis and gut sterilization with antibiotics, mainly vancomycin, in obese mice decreased the development of HCC. Additionally, senescence-associated secretory phenotype, a process occurring in normal cells in response to stress condition, has a crucial role in promoting obesity-associated HCC development in mice. And the next thing, that the activation of hepatic progenitor cells with the production of profibrogenic factors activate hepatic stellate cells and boost the production of collagen, which are the most important steps in cancer development. And the degree of hepatic progenitor cells activation seems to be correlated with the severity of liver disease and the risk of liver cancer. And finally, you can see that these progenitor cells were found in more than 50% of dysplastic foci or cancer nodules in the liver. So in conclusion, we can say that there is a difference in density, function, and composition of microbiota between healthy people and patients with metabolic abnormalities. Microbiota composition is relatively stable, but depends on genetics and also is modified by some environmental factors. In healthy condition, microbiota exert primarily beneficial effects. In pathogenic condition, altered microbiota plays a pivotal role in introduction and progression on liver damage. Microbiota appears to be involved in pathogenesis and also progression on liver cancer. And there is still not clear if this dysbiosis is the result of or the reason of metabolic syndrome, obesity, diabetes, and liver cancer. And at the end, I would summarize that I hope that all this information are true, because George Bernard Shaw said that beware of a false knowledge, it's more dangerous there than ignorance. And we also must remember we must support our bacteria, because as you can see, it is the only cartridge which some people have. Thank you very much.
Thank you very much. It was a very nice talk. I would like to have a question. Do you know about some trials which show that the modulation of the gut microbiome can prevent or ameliorate the NASH or obesity? If there are any clinical trials on this topic already?
Yeah, there are some clinical trials which were started not very long time ago, and it was really shown that just some of these new probiotics can slow or even resolve hepatic steatosis and decrease the NAS score. NAS score, I mean the inflammation, but there was no data about the decrease in fibrosis stage in this patient. But there are more and more studies, not only one probiotics, but probiotics with prebiotics and some others. So there are a few studies at the moment.
Yeah, please.
Congratulations on excellent report. I would like to ask you mentioned the leptin as a you mentioned leptin as an adipocytokine in the process of the insulin resistance and obesity, the fat tissue. Now it's a boom with adipocytokines. It's a lot of different adipocytokines. Something new about the role of these substances in induction, not only the changes which are connected with the metabolic syndrome, but also with malignancy. It's not only in the liver, also in the pancreas, for example.
Yeah, there are. Thank you for this comment. It's really true that adipokines now are a very hot topic, mainly not leptin and adiponectin, but some new ones, especially camerin, vaspin, visfatin, and omentin. Some of them seems to be protective, and some of them seems to be really harmful on the other side. And for example, camerin can have two faces. Camerin is important at very early stage of inflammatory process because it provoke NK cells to attack these pathogens. But when we have chronic inflammation, camerin is very, very bad adipokines because it maintains the inflammatory process. So that's true, and there are some new data and new research which try to assess the role of new adipokines in neoplastic diseases, but also in inflammatory diseases such as Crohn's disease, colitis, ulcerative colitis, and some others. But there are also some studies which also try to assess the role and to assess the association between psychiatric abnormalities and IBD, for example. We know that in patients with IBD we have psychiatric problem in many patients, and there are some studies which try to assess the role of new adipokines because some of them were found in the brain. So there are really, really broad spectrum of activity in the field of adipokines. And there are mainly, of course, checked in obesity, type 2 diabetes, and metabolic syndrome, but also in some other cases.
Thank you very much. Thank you very much.